This is Lawrence Bloomer at Morehouse College. The video that I've prepared here is about the colony-based sequencing database search. You should have this handout in front of you. This is a handout that's available, that your instructor should have made available to you, but it's also available on the Bean Beetle website. We are going to go to the Bean Beetle website to actually pull down data that were collected by students at Emory University, um, where they previously um, did PCR amplification of bacterial colonies that they picked. They did NBLAST searches and identified the bacteria that were picked from colonies. This particular handout describes using an Excel spreadsheet to do your analysis, that is, to do your community ecology analysis of these data. But in this video, I'm going to be using Google Sheets um, as an alternative, since the methods will be the same no matter what your computer platform is. In other words, Google Sheets will work the same on a Windows machine as it would on a Macintosh. So let's go ahead and start by going to the Bean Beetle website. That's here, and we'll go to, by the way, your instructor may have already pulled data down for you and made those data available, but if they didn't, you can go to microbiome database search and colony sequence database search. And then you have a number of choices. The choices that you make are going to depend on the kind of experiment that you were already doing. So let's imagine that you wanted to compare the microbiomes of beetles that came from black-eyed peas and compare those to compare those to the microbiomes of beetles that came from Idzuki beans. So under bean type, we want to select Idzuki and black-eyed pea because those are the two types of hosts that we're interested in comparing. We'll select adults, and you could select just one sex, but I think let's go ahead and select both sexes. We can do the analysis for one sex at a time later if we think that's interesting or useful. And then go ahead and submit this. This is going to leave everything else the same on this, and we see that we get 247 entries. These are these entries represent individual bacterial colonies that were picked by students at Emory. They did the NBLAST on those colonies after conducting polymerase chain reaction on them, and doing that NBLAST search on the Sanger sequence that was obtained, they determined what the genus was for each one of these colonies. We want to save these results. That's by clicking here on Download Search Results, and it's saving a CSV file. In this case, it saved it to my download folder. I suggest you give this a name that you can recognize. So I'm going to go ahead and open the folder, or rather open that file, and give it a new name. I suggest you do this as well. And so in this case, I'm going to rename this colony sequence black-eyed pea versus Idzuki, so that I know what it is. And rather than putting it in my download folder, I'm going to actually put it on my desktop where I'll be able to find it. Potentially, I'll be able to find it more easily. Okay, so that file's there. On my computer, it opened up as an Excel file. It may open up as a different kind of file on your computer, but it doesn't matter. Just give it a name. I'm going to click out of this. Now what you want to do is actually open this in, open the Google Sheets program. And so to do that, we need to go to a browser. So I'm there. I'm going to go to Google Sheets and open that. And you want a blank document. Okay, now go to File and Import. And it's taking me to my Google Drive. If you don't have access to a Google Drive, establish a Gmail account and create a Google Drive. This will be useful for you. I'm going to upload the data to my Google Drive. So I clicked Upload. Now I'm going to click Select File from my device. And I'm going to the desktop. And this is the file I just named a moment ago, Colony Sequence, Black Eyed Pea versus Adzuki. And it's a CSV file. I'll go ahead and open that. And now we need to tell it what those data look like. I'm going to replace the spreadsheet that I have open. The data are comma delimited. You can leave this selection to yes. It's not going to change anything. And click Import Data. There we go. So now we've got all of the data here. And what I suggest you do here is, well, several things. One is you could rename this spreadsheet. Right now it's untitled. I would call this spreadsheet Colony Sequence Black Eyed Pea versus Adzuki, since that's the nature of the file we have. Of course, you can give it any name you want. And the tab, I would double click on it and call it raw data. Then just click out of that. Okay, so you've saved the file. Presumably, well, right now this file is saved on your Google Drive. It's not on your computer. It's in the cloud. The next step you want to do is go to the tab, and there's a little down arrow. You want to click on that arrow and say copy to, and you want to copy this to the existing spreadsheet. That is, we want to create a new tab with an exact copy of all of these data. That existing spreadsheet should be this one, yes. And there's the new copy. I would rename this and call it reduced raw data. And then click out of that. Now what we want to do to the reduced raw data is actually clean up the file a little bit. So what we're interested in is we certainly keep these columns in place, the experiment ID and the sample ID. We're interested in the host because of course that's the comparison. We want to compare microbiomes from black-eyed pea to Adzuki beans. I would leave the sex there, but everyone's an adult, so this can disappear. We can get rid of that. We're not really interested in the type of auger plate that the colony came from or the phenotypic characteristics of the particular bacterium. I would go ahead and get that information out. So highlight those columns and go to Edit and say delete the columns. Delete columns, in this case E through J. Then we're interested in the identification. So here we have phylum, class, order, family, and genus. I would leave that, but the sequence, the sequence file, the PDF file, the AB1 file, those columns can also be deleted. So again, go to File, to Edit, and say Delete Columns. In this case, J through M. You don't need those. So now my reduced raw data file has the experiment ID, the sample ID, the host, the sex, and the identification of the bacterium. So at this point, I would say, well, it's already been saved, so you don't need to worry about that. Now we're ready to actually do the manipulation of these data. And that manipulation is going to be a pivot table manipulation. To do that, go to Data. Actually, I should say that what you're going to want to do first is I would highlight the entire data set. That's going to ensure that when you do the pivot table command, so we're going down to row 246 and we've got columns A through I. Go to Data and go down to Pivot Table. I would put it in this existing sheet. And click Create. Oh, I see what it's doing. Let's cancel that for a moment. Go to Data, Pivot Table, ask for a new sheet. Now, it's giving you some choices. It's created a new tab. That's what we wanted to do. And now we need to designate how the data are going to be actually organized. And so you're going to want to look at the right-hand side here, and the rows should be two things, sex and host. So you'll click on Add and you'll get a list of all the variables that you can choose from. The rows should be the host, and then click again on this and click sex. The columns in this case should be the information that gives us the identification of the bacterium. I would select genus. So look at what it's doing. It's creating a new table where the columns are the taxa, in this case the genera, and the rows are the treatments. It's Zuki beans and black-eyed peas, and then it's separating the two sexes. And then finally the values, the thing that we're actually counting in this table. The values are going to be the sample ID. In other words, how many samples, how many bacteria fit into each one of these categories. So go ahead and click that. At the moment, I would go ahead and use a filter. Let's make this simple and say that we want just one of the sexes. So there's two ways to do this. One is to actually filter by sex. So you could click on that and filter so that you just have females. We can do it that way. And now we've got a table. You can see it here where we're just looking at females. That is, microbiomes that were isolated from females, where the females either came from Adzuki beans or black-eyed peas. The counts that are shown here are the actual colonies and the genus that each of those colonies belongs to. You'll notice that there's one here in the beginning that has no identification, and that's because there were some samples that couldn't be identified. But before we do any manipulation on this, we need to actually copy this. And so I would highlight everything that's here and go to Edit and say Copy, and then what we want to do is create a new tab. So let's see, we can insert a new sheet. There it is. And then what you're going to want to do now is go to Edit and Paste Special, and you want the values only. This is really important. You want to just paste the values only. So now I have everything that was in the pivot table. So I'm going to click back on the pivot table. There's the pivot table we created. It's just for females from the 2 bean species. We have 146 bacterial colonies that were isolated. I've copied that entirely to another sheet as values only, and you can give this tab a new title. We'll call that Analysis. And this is the analysis of females. Okay, so far so good. Now, the grand total row we can delete. It's of no interest to us at all. So I would go there and say, whoops, not in Chrome, here in the sheet, and just say delete those values, or actually delete row 7. There we go. This column C, has no genus associated with it, so it's not giving us any useful information. I would delete that one as well. And so by clicking on that arrow, I can say delete the column. There we go. So now we've only got the columns in which we actually have information. Actually, let me just go back for a moment. I want to undo that. You know what? It would be better if we simply pick this up, because I want to change the total count at the end. So I don't just want to not have it appear. I want it to be deleted. So I'm going to go here and say delete the column. Oh, I see what's happening. So we don't have the totals now. So we're going to have to redo the addition here. Otherwise, we're going to be fooling ourselves, because remember I transferred the values, so the actual sums aren't being calculated here. So let's just, I've clicked on the grand total for this row, which is Itsuki. Oh, I see what's going on. We've got too much here. So we can delete this row. Not there. Here we go. Delete row 4. Let's delete row 5. It's just redundant. There we go. That's better. And here I would give an equal sign and say what we're interested in is the sum. So I would just type in sum and then put a parenthesis and highlight that row and then put an end parenthesis and then a return or an enter, and it's giving us the number here. And then do the same thing here, but you can save yourself some trouble. Just click on the upper cell which has that formula. You can see it right here, and grab the little box on the lower right and just drag it down. There we go. Okay, one more thing you want to do. The empty cells had no counts in them, so those should be zeros. Just enter zeros in those empty cells. So there are 2 there and there are 2 here. There we go. Now we're ready to do something interesting. So the first thing we want to know is how many different genera were present in each of these categories. In other words, how many different genera were present in the bacteria that were collected from females that were raised on adzuki beans, and how many different genera were found in bacteria collected from females that were from black-eyed peas. These grand totals are not the number of genera. These are the number of samples. So the number of genera would be the richness. So I'm going to type that in this column. And then what we want to enter there as a formula to actually do this calculation for us is a formula that's going to allow us to count the number of different genera that have non-zero values in them. And so we can do that fairly easily. That count it's the same command as we would use in Excel. It's COUNTIF. So give in that first cell for this row for adzuki beans, put an equal sign and type in COUNTIF, and then put a parenthesis, and then you need to indicate the range. So the range is that, whoops, let's just do it this way. I'm clicking at one and the other. Don't pick up the grand total. You're just interested in the actual values. And then, so you've got the range listed. The range is in cells C3 to cell L3. Then put a comma in. And what we're interested in is the counts. That is, how many categories, how many cells are in that count that are non-zero. So we want to indicate that the count should only include those where the values are greater than 0. So what you want to type in here is equals greater, sorry, not equals, but quote, greater than 0, end quote, and then put a close paren. Click enter, and it's going to give you the number of genera that were in this group. It's 8. Of course, you could have done it by hand because the set isn't that large. There's 1, that's not going to count. 1, 2, 3, 4, 5, 6, 7, 8 different genera. Then again, we can count the other one by just clicking here, grabbing that lower right-hand box and dragging down, and it turns out there are 8 different genera in both sets. So it looks like the richness, the number of different genera present for the microbiomes that were, that is the bacterial colonies that were collected from females from adzuki beans and females from black-eyed peas is not different from each other. The other thing that we wanted to evaluate is well, there's several things. This isn't in your handout, but I suggest you do it. The thing that's not captured by doing the richness calculation is what genera are actually dominant here. And really asking the question, are the same genera equally dominant? And I would just look at this and identify the genera that appear to be most common. And it looks like Enterobacter, at least in adzuki colonies isolated from adzuki bean females, include Enterobacter, Enterococcus, and Staphylococcus. I would make another little table, and you can do that by, we'll just pick up these values, the identifiers. I'm giving a copy command, and then I'm just going to go down and say paste those in. There they are. And then we can just identify those genera that we're interested in. So Enterobacter and Enterococcus, I'm just highlighting them and giving a copy command. And then we'll just paste them in here. And then Staphylococcus, I'm giving a copy command and pasting it in here. Okay, now what I want to do here is I want to calculate the percentage of the total number of samples. That is the total number, the proportion of the total number of counts that are represented by these three genera, which appear to be the most common genera overall. Though you can notice for black-eyed peas, it's not very common. Enterococcus is not very common for black-eyed peas, but it certainly is for the bacterial colonies that were isolated from beetles, female beetles that came from adzuki beans. So what do I want here? In this first cell, this should equal, and we're going to put a parenthesis around it, it should equal 35 divided by the grand total, which is 95. We'll put a closed paren, and I'm going to multiply it by 100 so I get a percentage. That makes it 38%. 38% of the bacterial colonies that were isolated from females from adzuki beans were Enterobacter. Again, we can get the same calculation for black-eyed peas by just dragging down. Let's do the same thing for Enterococcus. For Enterococcus, this calculation should be, I'm going to give a open parenthesis, it's Enterococcus, the value for Enterococcus divided by the total closed paren times 100. And then we can simply drag that value down and we'll get the next value, which is 1 divided by 49 times 100. It's only 2%. And then let's do the last one. And actually, I want to just double check here. Yes, this is doing what we wanted it to do because that value should be M4, and it is. Okay, for Staphylococcus. For Staphylococcus, again, we want this to be equal to the number of colonies that were identified for coming from females that were raised on adzuki beans. So that's that 39 divided by the total times 100. And hit enter, and then again you can just grab the little box and drag it down. So now we're seeing the relative proportions, and they're not the same, even though the genus richness was the same for both treatments, both adzuki and black-eyed peas. The composition isn't quite the same. You can see that there are more Enterococcus and Enterobacter in the colonies that came from adzuki beans than there were for the colonies that came from black-eyed peas. But for black-eyed peas, there's more Staphylococcus than there is in the colonies that came from adzuki. Okay, the next thing is we can calculate some diversity indices to see whether those are different between the colonies that came from adzuki beans and black-eyed peas. Again, the way to do this is to start out by grabbing all of the column labels except the grand totals. So for the actual data, I'm simply copying that, and then I'm going to paste it down here. Whoops, that's too much. Come on. There we go. And then I'll give a paste command. There we go. And then I want to get the row labels. So I'm giving a copy command and then a paste command. Okay. So now we have the rows and columns. I would do this once more while we're thinking about it, because there are two different diversity indices that we're going to calculate. So I'll give a paste command here, and then I want to pick up that first row again and give a copy command, and then I'm not sure why it's wanting to do that. Oh, come on. There we go. And now we'll give a paste command once more. There we go. Okay. So the first thing that we're going to do, and I'm following along with the handout. On page 13 of the handout, it describes the Simpson index. The Simpson index is a diversity index that incorporates both the richness and the relative abundance. And that particular index is described both in the handout, the formula is given in the handout, but I'm going to show you a slide set that your instructor may make available to you. This slide set is showing the Simpson index. It's calculated as the sum of the relative abundance squared for each of the taxa that are in your particular collection, in your particular community, where n is the number of individuals, in this case the number of samples of a particular genus, and big N is the total, the grand total for the number of individuals in a given treatment. So I'm going to leave the slide set for the moment. Let's go back to our sheet. That means this first cell and each individual cell here should be the individual relative abundances. And what we want to do then is actually calculate little n divided by big N for that particular row. We want to do this in the following way. So give an equal sign for the cell and an open parenthesis. The first value is C1. I would just type in C1 divided by, and then we want to use a technique that's going to prevent the value of the grand total from changing as we go through this. So you want to type in $ and then the cell of the grand total. So the cell of the grand total here is M3. So it's $ M3, and then close paren, and then you're going to hold down your shift key. This is at the above the number 6 key on your keyboard. So it's an up caret or an insert caret. It means make this a superscript and then a 2. Because what we want to do is square this. You'll remember, if we go back to our formula, it's little n, that is the actual count of a given taxon, in this case genus, so the number of individuals of a given genus divided by the total number of individuals of all the genera in a given treatment squared. So that's what we've done here. We've entered, in this case, the number of Acinetobacter that were in Adzuki, from females from Adzuki beans divided by the total number of individuals that were from females from Adzuki beans, and we're squaring it. Once you've typed that in, you can hit enter. So what just happened here? Oh, I typed in the wrong thing. It shouldn't be C1. It should be C3. Good. There we go. Okay. Now, I would click on this and drag across. Dragging across, it's having typed in that $, it keeps the value of the grand total the same for each calculation, which was what we want, but it will pick up the values of the uh, abundance of each genus in that row. So click on this and just drag across, and it's doing the calculation for us. And then once you've done that, then you can drag down. And by dragging down, it's picking up this other grand total, which is 49. We can prove that to ourselves by clicking in one of the cells, and we can see it's picking up M4 now, not M3. Okay, what can we do with this? Well, the Simpson index was the sum over all the values. So let's go back to our spreadsheet. Here I'm going to make this the value, rather this column is going to be Simpson, which is D, that's the variable, and all we want is the sum for each row. So this is equal to the sum, and that sum is going to be these values, and close the parenthesis. That's the Simpson index value for colonies collected from females from Adzuki beans. And if we click on that cell and grab that little box in the lower right and drag down, we'll get the calculation for the colonies collected from females from black-eyed peas. Notice these values are not terribly different from each other. But they're also a little difficult to interpret, and the reason they're difficult to interpret is because the way the Simpson index is calculated, larger values mean less diversity. And that's not very intuitive. So typically what we would do is we would calculate two other things, and those two other things would be either a reciprocal Simpson value or an inverse Simpson value. The reciprocal is 1 divided by D. That's the reciprocal. And the inverse is 1 minus D. So we can calculate both of those. So here the reciprocal is equal to 1 divided by the Simpson value, and then just hit return, and then we can just click on that and drag the little box down to get the other calculation. Again, these values aren't very different from each other, suggesting that the diversity is not very different between these two groups. And then finally we can do the inverse Simpson, which is 1 minus D. So again, give an equal sign, say 1 minus, again it's the D value, hit enter, and then drag down. The case is that for the reciprocal and the inverse, larger values mean greater diversity, but these values are not very different from each other, suggesting that the diversity is not very different between these two treatments. Finally, the last thing we can do, and what I've done here is I'm now at page 16 of the handout, is we can calculate the Shannon-Weaver index, or the Shannon index. I'm showing you the slide set called Introduction to Community Ecology. This slide set, by the way, is available on the Bean Beetle website. The calculation for Shannon is the negative sum of the proportion of individuals of a particular taxon times the natural log of the proportion of individuals of that taxon. The taxon we're talking about here could be species, but in this data set it's genus. Don't be frightened by this. It's simply little n divided by big N, the same thing we did before, and so that's how we can calculate it. We do the calculation a little differently, and that's because when you take the natural log of 0, you're going to get something undefined. So we don't want to include those 0 values in this. So the way we're going to enter the calculation is a little bit differently. We're going to say, and this is the same in Excel as it would be in Sheets. We want that to be equals if, and then do an open parenthesis, if this value, that is the actual count, is greater than 0. So in this case, if C3 is greater than 0, then put a comma in. So if we're saying if it's greater than 0, then do the following calculation. The calculation is calculate uh, the Shannon value, or the Shannon-Weaver value. So if the value in this cell is greater than 0, then take that value, which is C3, and divide it by, and again, we're going to use the same trick we did before. Use the $sign so that we keep that grand total value the same as we drag across. So we're going to divide by M3. So it should be $M3. Make a close paren. But then we want to multiply that by the natural log, that's ln, times, and then exactly the same formula, but you're going to need to use two open parentheses. And again, it's the same formula, C3 divided by $M3, and then two close parens, and then comma, and then put two quotes. The reason that you're putting two quotes and then a close parenthesis, the reason you're putting in two quotes is this is what the formula means. It's saying if this cell, C3, is greater than 0, do this calculation. But if it's not greater than 0, if it's 0, or it can't be less, but if it's 0, then just leave it blank. That's what we want it to do. Again, these double parentheses here are necessary for this calculation to be done properly. Once you've entered it like this, then hit enter, and it's giving us a value. Click on this and then drag across. That is, stay in that one row and drag across, and it's calculating the values. Notice where there are zeros, it's just leaving blanks. That's what we want. Once you've done that and you've got the whole row selected, grab that little box and drag down, and it'll do the same calculation for the other row. Now what we need to do is actually calculate the Shannon value. Remember, the Shannon value is the negative sum. So if this in the sheet is Shannon, then what we want that to equal is actually the negative sum. And again, this calculation is in your handout. I'm on page 17 of the handout. This should equal the negative sum. Put an open parenthesis and select all of the values in that one row, and then give a close paren and hit enter, and it's calculating the Shannon value. If you click on that again and grab the little box and drag down, you will get the Shannon value for the black-eyed peas. In this case, Shannon's actually a different, it's a similar calculation, but it's different, and it actually suggests that there's greater diversity among the bacterial colonies from black-eyed peas than there are from adzuki beans. Not very much difference, but a little bit, a little more diverse. Now, this calculation was done just with females from adzuki and black-eyed peas. You could do the same calculation with males to see if there's a difference between males and females. You could do the same calculation by not selecting sex when you pull down the data, and that way you would have males and females together, and you'd have a data set of 246. So you could do the analysis that way. These variables, the richness, the Simpson values, the Shannon values, the most common genera, and the percentage representation, all of those data could be part of the results in your report on a study asking whether there's a difference between the microbiomes that came from beetles from adzuki beans and the microbiomes that came from black-eyed peas. But keep in mind that these data are not the same as data that would come from the complete sequencing, the next-gen sequencing of the microbiomes that came from these beetles, because these data are based on picked colonies, on individual colonies that were picked from uh, cultured bacteria that came from the microbiomes of these beetles. This is going to end the recording I have on this analysis. It's worth doing this analysis and including it in your results, but there are other analyses, in particular the next-gen sequencing analyses, that you will want to do.